Hi guys, today we will be looking at photosynthesis, the topic that is really common on the exam and for some reason students don't really like it. So we'll try to make it as simple as possible. So to understand photosynthesis and obviously know where the stages are taking place, we need to quickly recap on the structure of the chloroplast. Chloroplast is of course an organelle where the process of photosynthesis is taking place. So this is the chloroplast. Chloroplast obviously got many adaptations. Has a double membrane, so that's outer and inner membrane of the chloroplast. So outer, of course, outside, inner more to the inside. Adaptation for the absorption of the light energy, obviously, it's hidden in the fact that chloroplast has a large surface area for the absorption of the light energy, and this is provided by the phycolloid membrane. Phycolloid membranes arrange themselves here in the discs, in the little stacks like this. This is the individual phycolloid membrane, and the stack of those individual phycolloid membranes arranged together we call granum. Granums are connected within the chloroplast through the lamella, which then leads to another granum, which is made of many phycolloid membranes. Remember, many means we've got increased surface area and we need that for the absorption of the light energy. Where the stages are taking place within the chloroplast, obviously, the first stage, the light-dependent reaction, happens in the phycolloid membrane, and the light-independent reaction happens in the stroma. So this is the stroma, this is the remainder, the fluid of the chloroplast. So imagine just like a cytoplasm. So that's called stroma. And this is where the light independent reaction, also called Calvin cycle, is taking place. So today we will look at the whole process of the light-dependent reaction, independent reaction, and see how they two link together. So light-dependent reaction happens in the phycolloid membrane. So I'm going to zoom in now this phycolloid membrane. So we are locating our process of the light-dependent reaction in the phycolloid membrane. So this is the phycolloid membrane. As we said, a phycolloid membrane has increases the surface area for the absorption of the light energy. So here we've got our source of the light energy, which is then absorbed by the chlorophyll. So this is the chlorophyll embedded in the phycolloid membrane. Inside of the chlorophyll, we can find a pair of electrons. A pair of electrons become excited once the light energy is absorbed. So those electrons now are going to leave the chlorophyll. So electrons has left the chlorophyll following the process of absorbing light energy. This is called photoionization. So those electrons now are going to pass through the chain of proteins embedded also in the phycolloid membrane of the chloroplast. So those are the proteins making ETC, electron transport chain, which will now be accepting the electrons. So remember, electrons previously got excited, they left the chlorophyll, and now they're going to jump from the carrier to another carrier. Those carriers are making electron transport chain. So they keep jumping and jumping and jumping. So imagine each time you will jump on the trampoline, you now are going to release energy. this energy is really useful because as much as we are working in the phycolloid membrane, outside of the phycolloid membrane, if you remember, we've got stroma. So all this space outside here, it's stroma. Within stroma, we've got hydrogen ions. Those hydrogen ions are going to use energy provided from those jumping electrons, and they will be actively pumped from the stroma to the inside of the phycolloid membrane. This is called chemiosmosis. So they jump here, increase now the concentration. If you remember properly, the whole process of the photosynthesis is to, of course, use water, carbon dioxide, the light energy, to produce glucose, hexose, oxygen, and ATP. You should remember from year 1, the ATP is formed using the enzyme ATP synthase. ATP synthase is also embedded in the phycolloid membrane. For the ATP synthase, the main job of the ATP synthase is, of course, to form ATP. For this reaction to happen and for the enzyme to become activated, we need hydrogen ions to pass through the ATP synthase. Passing through this ATP synthase, the shape of its active site will change and allow the formation of ATP to happen. So this is how ATP is produced here. Now we need to come back to our diagram, to the beginning, to the starting point. Remember, once the photoionization took place, so once the light energy was absorbed by the chlorophyll here, the pair of electrons got excited and they left this chlorophyll, leaving it empty. So for the process of light-dependent reaction to continue happening, we now need to replace those electrons. The source of the electrons we will find in water. So photolysis of water will take place. We're simply saying water now will be broken down to donate hydrogen ions, pair of electrons, and oxygen. So what we can gather from this process here of the photoionization of water is we now got source of the electrons that will simply replace the ones that we've lost in the chlorophyll because of the photoionization. Now we also received here the hydrogen ions, and which can further increase the concentration in the phycolloid membrane. Hydrogen ions will have one more function, which I will talk about in a second. And we also achieve oxygen. Coming back to the equation of photosynthesis, you can see the oxygen, together with ATP, of course, are the products of photosynthesis. So far, looking back at the equation, we've used water in the light-dependent reaction. We've produced oxygen in the light-dependent reaction, and we also produced ATP in the light-dependent reaction. Looking at the diagram, it's almost completed. However, you need to remember about those electrons. Those electrons are jumping and jumping and jumping. So imagine like you would jump all day long on the trampoline. Obviously, you're going to run out of energy. So those electrons, eventually, we need to accept them, because those electrons obviously cannot continue jumping throughout the ETC, because each time they jump, the level of energy released will decrease. So the electrons now will be accepted together with the hydrogen ions. So electrons and hydrogen ions will be accepted by the coenzyme, sorry, that meant to be plus, NADP. Okay, so remember P for photosynthesis, because in respiration you've got similar coenzyme for NAD. So electrons, which were excited previously from the chlorophyll, and hydrogen ions now will be accepted by NADP, forming reduced NADP. Also, you can just write this down as NADPH. Where the reduced NADP is coming from and what's the idea of being reduced? Remember, reduction is the process of gaining electrons or hydrogens. Hence, our NADP coenzyme become reduced because it accepted both electrons and hydrogen ions. So looking back at our whole process here of photosynthesis, we've got main products. We've got electrons here from the photolysis of water, but they've been used. They've been used to, of course, replace the electrons which were excited from the chlorophyll. The main products then of the light-dependent reaction leaves us as oxygen, which again comes as a product of photolysis of water, ATP, which we produced here thanks to the help of the ATP synthase, and reduced NADP. Those are our three main products of the light-dependent reaction. So to summarize this process one more time, we started with the photoionization of the chlorophyll. The light energy was absorbed by the chlorophyll, and the pair of electrons found in the chlorophyll got excited. The pair of electrons then were transported throughout ETC, electron transfer chain proteins, which kept accepting those electrons. Electrons were jumping from carrier to another carrier, releasing energy. That energy was then taken by the hydrogen ions in the stroma, so they could be actively transported to the inside of the colloid membrane, increasing their concentration gradient. Hydrogen ions had a really important role because they needed to pass through the ATP synthase to change its active site. Thanks to this process, the reaction of the formation of ATP could be catalyzed. The problem with lost electrons in the chlorophyll was sorted thanks to the water, which is one of the substrates needed for photosynthesis. So water was broken down, photolysed to the hydrogen ions, which further increases the concentration of them, electrons, which replace the electrons lost in the chlorophyll, and oxygen, which is of course the product of photosynthesis. The jumping electrons and hydrogens, which increase the concentration, were then accepted by coenzyme NADP, forming reduced NADP. Why reduced? Because it's gained the hydrogens and electrons. So far, throughout the equation of photosynthesis, we've used water, we've produced oxygen, and we also produced ATP. So you can predict now that the other missing parts of this equation, as carbon dioxide and hexose, will be then seen in the second part of photosynthesis, which is called light independent reaction. Light independent reaction will show us how those are used and produced, but also will show us how two of the main important products of photosynthesis, which is ATP and reduced NADP, will be useful. So we will move now to the light independent reaction. Hopefully you remember where the light independent reaction is taking place. is taking place in the stroma of the chloroplast. So as we've said, we will need carbon dioxide for this reaction. Carbon dioxide obviously comes from the atmosphere through the stomatas. Carbon dioxide contains one carbon, hence I'm going to draw here one dot. So the dots I will be drawing correspond to the numbers of carbons, so that's one carbon. Light independent reaction, also called Calvin cycle, as any other cycles, has to start and finish with the same kind of molecule here. And what is waiting here and starts the process of the Calvin cycle is ribulose biphosphate. Ribulose biphosphate. Ribulose biphosphate is also a carbon molecule, and this carbon molecule this time has 5 carbons. So we've got here 4, 5 carbons. So one more time, it's a cycle, so each cycle has to begin and finish with the same product here. So we're starting with a ribulose biphosphate, which has 5 carbons. So ribulose biphosphate in the stroma now will react with carbon dioxide, and this reaction is catalyzed by enzyme called rubisco. So once they join together, as we can obviously predict, we will get a 6 carbon molecule. However, this 6 carbon molecule, it's not a stable molecule, hence straight away we are achieving 2, instead of that, we're achieving 2 3 carbon molecules. So I'm going to remove this 6 carbon molecule because this is not the part, but instead, obviously, we are achieving 2 carbon molecules, and those are called glycerate 3-phosphate. Okay, so RUBP, ribulose biphosphate, combines with carbon dioxide from the atmosphere. This reaction is catalyzed by an enzyme rubisco, which gives us 2 molecules of glycerate 3-phosphate. This glycerate 3-phosphate is going to be now reduced to 2 molecules. Those molecules are called triose phosphates. And if you can look at the maps properly, they still have 3 carbons each. How is it possible to be reduced? Obviously, we look at the reduction before. Reduction is gain of the hydrogen's electrons. This is where our products from the light-dependent reaction will be quite useful. As we can see, we've got here the reduced NADP, which carries hydrogens, and that's its job. That's the job of the coenzymes. So our product from the light-dependent reaction, reduced NADP, will be essential here. Reduced NADP now is going to give out its hydrogens and electrons, so it's losing them, and if it's losing them, it becomes oxidized. So those hydrogens now will be used to reduce, just pick different color, to reduce GP to TP. This process also requires energy, and as we remember, we've produced ATP in the light-dependent reaction, hence it's useful now, and it can help out with the reduction of GP to TP. So now we are almost at the end of the Calvin cycle, and we've got 2 triophosphate molecules. As I've mentioned before, the cycle has to finish and start with the same molecule. So triophosphates now will have 2 jobs. Job number 1, it will be to regenerate our initial molecule here, starting the Calvin cycle, which was ribulose biphosphate. So coming back to our equation of photosynthesis, we can now tick off carbon dioxide because we have seen it. However, we are now interested in hexose. And this is the second job triose phosphate will need to do. Triose phosphate will need to contribute and donate the carbons to produce hexose. The main important job of triophosphates is, firstly, of course, to regenerate RUBP. This is really important job because without RUBP, the further Calvin cycles would not happen. So first possible carbons that will be formed here will be donated to regenerate RUBP, and follow-up carbons, remaining carbons, will contribute to, of course, produce hexose. To summarize quickly Calvin cycle one more time, we started the cycle with the molecule ribulose biphosphate, which contains 5 carbons. RUBP, ribulose biphosphate, now reacts with carbon dioxide in the stroma, and that is catalyzed by an enzyme called Rubisco. This reaction allows the formation of 2 GP molecules, where each of them contains 3 carbons. GP molecule will be now reduced to triose phosphate using the products from the light-dependent reaction, which is reduced NADP and ATP. Reduced NADP will now donate hydrogens and electrons to reduce GP to TP, and also this reaction requires energy, hence we need ATP for that as well. Triose phosphate will now be used to, number 1, regenerate RUBP so the cycle can continue. Number 2, to produce hexose. In that way, we now can summarize our equation and add on some labels. So, of course, carbon dioxide has been seen and used to combine with RUBP in the light-independent reaction. So was the hexose produced in the light-independent reaction, or Calvin cycle, if you like. So that's the whole process of photosynthesis. And of course, the tips are here. Not only content is important, but the past paper questions. So become confident with the questions. Make sure you practice many of them, including the practical skills. So that's everything for photosynthesis. See you later.